All right, what's up, you guys? So in this video, we're talking about neurotransmitters. And neurotransmitters is a really good name for what they are, right? They're released by neurons, and they transmit information to the next cell. So we're either talking about like an example of a neuron sending a neurotransmitter to another neuron, right? That's the example we're going to illustrate over here. We're talking about just neurons. You can also, though, have a neuron sending a neurotransmitter to a muscle too. That's how we control our muscle cells. It's how we connect the nervous system to the actual muscles that are moving. Cool, so let's go and talk about them. We kind of have two categories. So one of them is excitatory. So that's going to tell the next cell, oh, hey, you should activate, you should fire an action potential, right? So I'm going to jot this down. Excitatory makes the cell more likely to fire action potential. Right, on the other hand, we have inhibitory neurotransmitters, and these make the cell less likely to fire an action potential. Cool. Let's go and talk about them one by one. Let's talk about excitatory. Right, so we already talked about they're going to make the cell more likely to fire an action potential. Does that mean you're bringing the cell closer to thresholds or farther from thresholds? If you have a solid understanding of action potentials, you realize that you have to reach threshold to fire it, right? So you're going to bring them closer to thresholds. Excitatory neurotransmitters bring things closer to thresholds, right? More likely to fire an action potential closer to threshold. Does this mean that you're depolarizing the cell or you're hyperpolarizing the cell? Right, check in with yourself. Do you know this? Closer to threshold, you're depolarizing the cell. Just double checking again. Does this mean you're making the cell more positive, or does this mean you're making the cell more negative? What does depolarizing mean? This means you're making the cell more positive, right? Neurons are already very negative. To cause that change, the exciting change to fire an action potential, you're making that cell more positive. This will help us answer the question, what ions do they allow into the cell? Right, they're going to let in our excitatory neurotransmitters. Are they going to let in cations? Or are they going to let in anions? Right, we just talked about the logic of how we know we're going to make the cell more positive, so it makes sense they're going to let in cations. Right, so the specific cation that excitatory neurotransmitters let in is they let in sodium. Cool. And what's an example of an excitatory neurotransmitter? You only have to know two for this class, so let's talk about these two. The two that you need to know are acetylcholine. Right, and can you think of the other one? It's a good moment to quiz yourself. Glutamate. Cool. I'm gonna erase this stuff over here. A quick note on acetylcholine is the only neurotransmitter used on muscles. Right, so that's kind of a weird idea that we can only use neurotransmitters to excite muscles, to turn them on, to tell them to fire an action potential. So you're probably wondering, well, then how can we turn them off if there's no inhibitory neurotransmitter for muscles? We'll talk about that in a different video. Cool, so just to draw it out, let's say this cell fires an action potential. This neuron fires an action potential. It releases a neurotransmitter. Let's say it's glutamate. Wow, my bad. Glutamate. It releases glutamate. It binds to this cell. Then this cell will allow sodium to come in through its channel. That makes the cell more likely to fire an action potential. It makes the cell more depolarized, right? So glutamate makes the postsynaptic cell slightly more depolarized, so slightly closer to threshold. Cool. Not all neurotransmitters, though. Fix this in glue. Not all neurotransmitters, though, are excitatory, right? We also have inhibitory ones, right? They're going to make the cell less likely to fire an action potential, so they're going to bring it farther from thresholds. So are they going to depolarize a cell or are they going to hyperpolarize a cell? Right, I'm going to ask you the same set of questions. If you didn't get them before, this is your chance to redeem yourself, right? So farther from threshold, are you depolarizing or hyperpolarizing the cell? That means you're hyperpolarizing it. Hyperpolarizing, does that mean you're making it more negative? Or does that mean you're making it more positive? Right, check in with yourself. Do you actually know? Right, a cell that's less likely to fire an action potential is more negative, farther from threshold. So you can make a good guess about what ions do they allow into the cell. Are they going to let in cations? Or are they going to let in anions? It's up to you to think about it. Right, to make the cell more negative, you have to let in anions. Right, so the specific anion that they let in is chlorine. So they let in chlorine. Cool. Right, so, oh, then we have the two examples, right? Sorry, I almost forgot. We have two inhibitory neurotransmitters to know. The examples that you guys need to know are GABA and glycine. Right, so notice that glutamate and glycine low-key sound a little similar, just understand that they're different and memorize which one is excitatory and memorize which one is inhibitory. Right, so like the same deal, I'm gonna kind of illustrate it. This cell could fire an action potential. Once the action potential reaches the axon terminal, that could cause this to release GABA into the synapse. GABA would bind to a receptor on the postsynaptic cell, a ligand-gated receptor, open it up, and then allow chlorine to come into the cell. Right, so sometimes you can get two conflicting signals, right? It's up to the neuron to decide. Quick little quizzing moment is which part of the neuron decides to fire an action potential? Right, I'm quizzing you guys on this because now we're at the point where we understand, like, oh, this one neuron here is getting conflicting signals from two different neurons, right? This neuron is saying go, this neuron is saying stop. So we have to summarize the information kind of right here, right before the axon, right? The action potential happens along the axon, so you have to summarize it right there before. This is at the axon hillock. So just a quick moment of quizzing you guys. Since there is so much information in this class, you should be quizzing yourself, right? I just read a good thing where it's like, you actually learn. You don't learn by putting information into your brain. So you don't, like, reading helps, but what you can do to learn the best is to retrieve information from the brain. So practice quizzing yourself more than you practice reading, right? If you quiz yourself 3 times on the same thing, that's more effective than reading the same piece of information 3 times. Okay, cool. So let me know if you have any questions, and I'll see you in the next video.